enrollment or inclusion criteria. Ages 50 to 85. Ongoing AD treated with donepezil. Very interesting here. The MMSE of 12 to 24 inclusive. Very interesting. At screening. And 12 or 10 to 26 at baseline, so they allow a little variability. I don't know what a Hachinsky ischemia score is. Ischemia, of course, is like a blockage. So if there's a stroke, I guess post-stroke. Now, there's really not much inclusion criteria. Not too much in the inclusion-exclusion. Usually, it's a much bigger list. Go figure. All right, so study sites. Looks like they have 197 study sites, which is pretty interesting. We'll look at those in a second. And let's write down the NCT code for faster access. Okay. And let's actually look. So now the study director, we should write that down. We get a list now of 4 of the company's employees. Let's look at the 197 study locations. So we've got, let's add up how many are in the U.S. 1, 2, 3, 4. Well, it's interesting that they're numbered, isn't it? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 11, 12. A lot in Florida. Guess there's some Alzheimer's there. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 and 12 is 24. 3 more in Georgia, 2 more in Louisiana, makes 5. That's 29. Another 2, 31. Another 2 here, makes 33. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6 in New York. And you'll see why this is actually very important in a second. 2 more in North Carolina, and 4 more in Ohio makes 6 more. So 45 and 6 is 51. 1 from Oregon and 4 from Pennsylvania makes 5. 5 and 51 is 56. 2 in Texas, 1 in Utah, 1 in Virginia, and 1 in Washington are 5. So we've got 61 in the U.S. Let's look. This is strange, huh? I have a lot in Argentina. $17 in Argentina. Strange. I would imagine there would be no clinical trials in clinical trial sites in a normal study in Argentina. 6 in Australia, which sounds about right. 4 in Bulgaria. Somewhat diversified. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 in Canada. 4 in Chile. 1, 2, 3, 4, 5 in Croatia. I'm guessing this is the Czech Republic. 5 in Czech Republic. 4 in France. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 in Germany. 2, 3, 4, 5, 6, 7, 8, 9 in Italy. 3 in Korea. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 in Poland. 1, 2, 3, 4, 5, 6, 7 in Serbia. 2 in Singapore. 1, 2, 3, 4, 5, 6 in Slovakia. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 in Spain. 3 in Taiwan. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17 in the UK. Let's see if this adds up, by the way. It does. All right, so the two biggest countries are, of course, the US, Argentina, the UK, Canada, and Poland, and Germany. It's about right. There's nothing untoward. What you're often seeing in some clinical trials is there's a weird geographical distribution that results in poor quality data, and it looks as if this company did a very good job of ensuring that that won't happen here. These are all kind of first world countries, especially the largest ones. I'd say that Argentina is a little out of place, and I mentioned that earlier, but other than that, it's a relatively good distribution. And I worry sometimes when I see someone doing all their clinical trials in Russia or something like that. So it looks pretty good to me. All right. So that's really it here. Everything's set up there. A drug has to definitely gets approved in some indications and not others, for sure. And so we know the primary endpoint is this co-primary endpoint. At least we think it's a co-primary endpoint, but we don't know for sure. So the question on a primary endpoint is, do you analyze them sequentially or simultaneously? And so if you analyze them sequentially, basically you're saying the first primary endpoint has to be met, and then the second primary endpoint has to be met afterwards for the trial to be successful. If you analyze them simultaneously, you could have a mixed result. You could have one meet its primary endpoint and then one not meet the primary endpoint. And that's why the word primary endpoint is a little strange. They're kind of co-primary endpoints, and there's sort of two ways more or less of doing it. So we don't know what the statistical analysis plan is, which is called the statistical analysis plan, SAP. And unless we had the exact protocol for the clinical trial, we wouldn't be able to determine what the SAP is. We can listen to the company, but companies can often be somewhat misleading with respect to their SAPs. Okay, so before we get to the next clinical trial, or press release, I should say, one of the things that I would look at is I'd look for information on interbreeding. So one of the best places to go might be PubMed. PubMed is sort of the clinical trial, or is the medical information scholarly published journals repository. So I'll look for any published information on interbreeding. Now, if you don't have a sort of access to these articles through a database, you can actually use, and I would never advocate you use it, but it's something called Sci-Hub. So you go to sci-hub.cc, and you can actually pull some of this stuff. And I don't think you should use it, but it's something that you could certainly think about. So let's read some of this stuff. So both of these papers seem to be review papers that address more than just more than just interberdine. So here, enhancement of the acetylcholine response using 5-HT6 receptor antagonists, serotonergic neurotransmitter. I've said that word a zillion times, but it's been a long day. Is impaired, neurotransmitter system is impaired as AD develops and progresses. Modulation of this pathway is therefore considered to be of therapeutic value. Serotonin 5-HT activates specific 5-HT receptors consisting of 7 distinct classes, 5-HT1 and 5-HT7, based on their structural and functional characteristics. The 5-HT6 receptor is expressed primarily in brain areas involved in learning and memory processes, the cortex and hippocampus. 5-HT6 receptor antagonists are thought to enhance cholinergic neurotransmission. Iloperidine, interesting, is an orally available 5-HT6 antagonist and showed promising efficacy and safety data in phase 2 trials. Although iloperidine is safe and well tolerated, however, iloperidine did not meet its primary efficacy endpoint versus placebo in 2 recent phase 3 trials. Eteperdine is another 5-HT6 antagonist, so now we know what the mechanism is. So when we go to eteperdine, we can write mechanism 5-HT6 antagonist. Great. And if you don't know what an agonist or an antagonist is, I suggest you look that up and learn a little bit about pharmacology. You would need to know that to analyze something like this. All right. That is currently in phase 3 clinical trials in patients with mild to moderate AD already on donepezil. So there's 2 clinical trials entries here. We have Mindset, which ends in 934, but we don't have this one. So let's take a quick gander. I think it's the open label extension study. So I don't want to... Yeah, it's a 12-month open label study. So basically after you go into the Mindset, you can do the extension study. Analysis of data from phase 2, evaluation of treatment with eteperdine indicates that addition of this treatment to donepezil may improve the cognition and function of patients with mild to moderate AD. So again, I would save this. Let's save this file in here. Let's make a folder called Axon, and you can save all of your data together. And again, this paper is free, so there's no issue in saving it. I just like to save these by their title: drug candidates in clinical trials for Alzheimer's disease, Hung et al. No relation to the company's CEO. And maybe put the date, 2017, I'm guessing. Yes. Okay. So there's some histamine antagonists as well in clinical trials. And there's nicotinic drugs, amyloid, of course, glutamate, and so forth. And if I were you, I'd read this entire paper. Now, what's interesting is that they have some references here. And I think we want to get those references. So, oops. Let's see. Let's look at some of these references. Doesn't look like they have a whole lot here. We'll print this out and go back to it, but it has the Axivant website as one of the press releases, as one of the references, which I don't think is going to help us too much. So that's fine. Let's look at the second one: emerging chemical therapies targeting 5-HT in the treatment of Alzheimer's disease. Where's the PDF? Get access. All right. I'm unable to get access through this account, so I'm going to pass on reading further, but that's fine. So there's PubMed, but there's also just a quick Google search. Interberdine. Let's take a look. And I quickly see here that there are some synonyms which strike me as surprising. GSK 742457. What's that? I know GSK is GlaxoSmithKline, one of the biggest pharmaceutical companies in the world. So I wonder, did this drug come from them? GSK 742457. Let me Google that. Huh, look at that. Axivant banks on GSK's scrapped project. So for anyone who doesn't know, I actually was bidding on this as well. And in fact, I bid $5 million as well as Axivant. So, just so you know, I know quite a lot about this. But I'm pretending that I'm seeing this for the first time. And I had nothing to do with this. So you can see here from this article that it looks like Roivant scrapped the project, bought the drug from GSK. So that's kind of interesting. So that's very important. So one of the things you want to always ask about a drug is, where did it come from? So now that I can just Google GSK 742457, and one of the first things that comes up looks like a published clinical trials. Okay, so let me save this. 2 randomized controlled trials of SB 742457. Mild to moderate Alzheimer's disease, Maher Edwards et al. 2015. All right. 2 previous studies of 457, a 5-HT6 antagonist, suggest the efficacy of improvements in cognition and global outcome in Alzheimer's disease. Look at that. So we had study 1 and study 2, and they were both about 600 patients, which is interesting because Mindset is 1,300 patients. So let's make room for this. Phase 3, study 1, in Alzheimer's, N equals 576. And phase 3, study 2, in Alzheimer's, N is 684. Okay. And study 576 investigated 457 and donepezil as monotherapy, and this was an addition. So both studies failed to achieve formal statistical significance for the primary objectives. Okay, well, that's not good. .457 monotherapy was not statistically significantly different from placebo on any endpoint. 35 mg showed statistically significant differences relative to placebo for ADAS-Cog at weeks 12, 24, and 48. Stat sig on ADAS-Cog at weeks 12, 24, and 48, but not 36. Okay, that's fine. And let's see, for ADCS ADL for weeks 12 to 36, but not week 48. All right. So sort of mixed results, huh? We'll get the raw data in a second. There's a lot of data here that's going to be really important to print out and read. There's a donepezil active control here and 15 mg and 35 mg. So let's write that down. 15 mg iteperdine, inteperdine or iteperdine, inteperdine versus 35 mg inteperdine versus donepezil versus placebo. So this is a pretty complicated study. So there's an NCT number for this as well. So let's put that down. And there's an NCT number for study 2. So we'll look those up in a second. Okay. Study 1 was conducted from 2008 to 2010. And study 2 was conducted roughly at the same time. I'd like to see the results. Here we go. So it looks like at week 12. So weird. Okay, let's make a... So it's pretty simple. There's 4 groups, right? Placebo, 15 mg iteperdine, 35 mg iteperdine, inteperdine, and donepezil. And there's a primary endpoint, which is 8S-COG, right? So 8S-COG, and there's 2 time points, week 12 and week 14. And 8S-COG is the kind of scale that the lower you go, the worse. So after 12 weeks, placebo went down 0.1, drug did not change, and inteperdine went down 0.2. And I'm sorry, 8S-COG is the higher it goes, the worse it is. So it looks like donepezil patients did the best. And week 24, placebo is declining, 15 mg getting better, inteperdine's getting better, or getting worse, and donepezil is getting better, is the right way to interpret it. Let's look at the 8S-COG scale while we're at it. Tasks measuring the disturbances of memory, language, praxis, attention, and other cognitive abilities, which are the core symptoms of Alzheimer's disease. This is the Slovak version validation. Let's maybe go somewhere else. "11 parts, takes 30 minutes to administer. Word recall task. Naming objects and fingers, following commands, shapes, letters, orientation, words, comprehension, etc. So I guess the greater the dysfunction, the greater the score. So the greater the dysfunction, the greater the score. All right, so placebo is going down, donepezil is going down, but inteperdine is going up. That's a little strange, I'd say. It's quite strange, quite strange. Not good, right? Not good at all. Let's see how it did in study 2. There's study 2. We had placebo, 15 mg and 35 mg, and no donepezil active control. Everyone's on donepezil, right? So let's copy this, paste it, a little copy pasta. We got week 24 and week 48, no donepezil. Let's see what the data is here. Let's see, so placebo goes up 1.2 at week 24. And you can see in the other study, it only went up, it went down 0.3. So it's kind of funky because these should be the same, basically, right? Although in this study, the patients are on background donepezil. So it's a little strange. Let's see, on 15 mgs, it went up 0.5. And then on 35 mgs, it went down 0.4. So what we'll do, I guess, is subtract from placebo. So here, placebo is getting better, and we're getting worse. That's pretty bad. And here, donepezil is actually improving. So a negative score would be better, right? This is worse. It's a point worse than placebo. Let's look at 48 weeks. So you've got 3.4 for placebo. So these patients are getting really sick. At 15 mgs, you also have 3.4, so the same. But at 35 mgs, you have 1.8. So here, patients are doing much better. And here, they're not changing. So you got one study, basically, that shows 35 mgs intepirdine is pretty good. And you got one study here that shows it's worse than placebo, which I think is a little strange, needless to say, because that's what they have to prove in the next study. So it's definitely not good that they saw that. A lot of variability, obviously, in clinical trials, but it is kind of strange. So then you have the second piece, which is the CDR sum of boxes, or CIBIC, is another way to do the clinical trial. So I'm going to copy paste again, kind of do the same analysis except doing this scale, which is the CIBIC plus. All right, so the CIBIC plus, patients in placebo had a 3.9 at week 12, a 4 at 15 mgs, and a 3.9 at 35 mgs, which, as you may remember, is the dose that they're going to use in Mindset. So that's kind of the dose that we're going to always be looking at. And then 3.7 for donepezil, which is also called Aricept. Okay, so after 24 weeks, they had 4, 4.2, 3.9, and 3.7. And so I don't know how CDR sum of boxes works. So we should go, it's not CDR sum of boxes, this is the CIBIC plus. So let's look at what that is real quick. CIBIC plus scale. All right, so in the CIBIC plus scale, it's a 7-point scale. 1 is improved, 7 is worse. So you want to go up on the scale. You don't want to go down, right? So higher score is better, in essence. So placebo patients were 4. I'd like to know the change, what the baseline was. I doubt this is a change. This is a mean. So maybe they have baseline here somewhere. Doesn't look like it. All right, because it's hard to know if you don't know what the baseline is. This could be changed though. Either way, you can see donepezil seems to be outperforming intepirdine but really tough to tell, and the p-values are bad. So let's take some of these p-values down as well. So this was almost statistically significantly worse. It looks like this was nowhere, and this was also nowhere. All right, so study 1 was like a total bomb for these guys. So let's look at CDR sum of boxes. In CDR sum of boxes, we had minus 3.2 for placebo at week 24, and minus 5.5, so they're getting worse, for placebo at week 48. And here you have minus 1.9 for 15 mgs and minus 1.3, growing to minus 5 and minus 3.5. So definitely a slowdown. It looks like a slowdown in the progression. Now, if you're a cynic, you would say that, well, I think if you're an optimist, you'd say that the clinical trial, I don't know, that the clinical trial was, see, they cut off the p-values here, which is a little annoying. You would say that the clinical trial is this was the well-done clinical trial and this was the poorly done clinical trial, but a priori there's no reason to assign which one was done correctly and which one wasn't. So there's more to sort of think that through in a second, but let's get these p-values. So this p-value was statistically significant, 0.012, but that's not a great p-value. And this p-value was not statistically significant, and this one a little confused here. Hold on. I'm not looking at the right secondary endpoint. See, I grabbed this data, but I should be grabbing this data. So let's redo the CDR sum of boxes. So it's 0.9 for placebo, 0.8 and 0.7. And here it's 1.6, 1.9, 1.5. And the p-values are 0.0711 and 0.462. So CDR sum of boxes did not go great. And let's look at how you analyze that. Let's see if they have that here, CDR. So this is higher is better. And it's sort of all over the place, as you can tell. A sample CDR table, let's see if... Now the co-primary endpoint now is ADCS-ADL. So keep that in mind. There's no p-values for this, but you can see the drug doing a little bit better, maybe not, than placebo. And then here, you can definitely see the drug doing better than placebo. There's a repeated analysis change you can do. And that's a way to sort of measure the study over time. And then there's a mild to moderate stratification, which again I'm not sure I believe in. too well. Here's some visual representations. Change in ADAS-Cog. The darkest line is placebo, and the box is our dose. So you can see here, there was a trend against improvement. Here there was a trend for improvement. That was study 2. So I'm guessing column 1 is study 1, column 2 study 2. These look about the same. Here, placebo and drug are about the same. It's really it's hard to tell the difference. Here, again, in study 2, the drug separates. This is a more well-run, maybe, study. If you believe that the drug works, this study was well-run, this study wasn't. But again, there's no a priori reason to believe that. So we got our first taste of what this drug is all about, and it looks like a pretty ugly situation in the sense that study 1, if you can find a reason to deny why study 1 is relevant, you could look at study 2 and get a little bit excited that you would get a little bit of activity. And again, I can't speak to my own experiences in this process. I want to look at this as objectively as I can from a sort of a neutral perspective. Again, pretending like I've never heard of this drug before. So, pretty weak efficacy, just 1 point or so better on ADAS-Cog, and the p-value of 0.01 is not too impressive. And then again, when you include study 1, you really get very worried about the results here. So if you were to, let's say, buy some puts in Axavanel, I can show you, you can short the stock, and if this study failed, the stock would likely go down, I think I mentioned about 75%. So let's take a look at, again, the stock price is $20.35. Let's go to Yahoo Finance since everyone has access to that. And type in Axon, type in options, and let's see, let's go to the October options. And if I bought puts, let's see, the 15 puts, I think the stock's going to go to 5. The 15 puts would be worth $10, so you double your money, if that makes sense. So this is like a lotto ticket, basically. You double your money if you're right. So that's sort of the way this would work. But if there was a real effect, and again, we've only seen 2 prior studies. We know that this study was 684 patients, and that this study is going to be, the mindset study will be double that, more than double that, 1,300 patients. So, but I think the best way to look at clinical trials is you look at past clinical trials. In the absence of that, you can look at some of the target data and preclinical data, but they've run experiments, and these experiments were not very good. This experiment was pretty good. Drugs don't work at low doses, and they'll work at higher doses. That's very common. So I believe that you can discard the 15 mg doses from both studies. Basically say that this is too low, right? If you take a speck of dust of Tylenol or Advil, you're not going to get a response. If you take more than a speck of dust, if you take enough specks of dust, say this many milligrams of specks of dust, you may get a response. Now, the problem is that we did not see that 35 milligrams was successful here in this study 1. We did see that it was successful in study 2. Now, in study 2, all patients were on donepezil, so the theory could be that maybe somehow combining these two would be beneficial, that there would be some sort of synergy, but I don't know. And the fact that the secondary endpoints didn't do well is a little concerning too. So There's, I think, more studies of this drug. I'm not sure, though. But let's sort of take a break from here. This is sort of the first few hours of how I would look at it. And again, the signs are not good. I try to reserve judgment just because I don't want to have a preconceived notion as more information comes in that I want it to influence my final decision. But I would say off the bat that, typically if you run an experiment and it fails and you run the experiment again, it fails again. And you can change things, obviously, to maybe pick the population where the drug does the best in. But I think that we have a lot to sort of think about here with the failure of this study 1 and the success of this study 2. But again, maybe the company can explain why we should discard these results and believe in these results. So we'll have more. This is a very, very preliminary analysis. We got way more to do here, but I hope you enjoyed this first session, and we'll be back with more later.